Meiosis. You know, meiosis is a reduction division, and it occurs in plants during gametogenesis, and in animals it occurs during sporogenesis. You can say that meiosis is a nature's way of inducing variations and keeps the chromosome number constant generation after generation. Meiosis. Meiosis produces genetically different type of cells. All the four daughter cells produced by meiosis are genetically different from each other and also different from the mother cell. As I told you, that this is a nature's way of inducing variation. Meiosis includes two divisions of a cell, that is, Meiosis-I and Meiosis-II. Meiosis-I is heterotypic division or reduction division. It leads to the reduction in chromosome number, that is from 2n to n, that is from diploid to haploid, and segregation of homologous chromosome occurs. Stages of Meiosis-I. Prophase-I. Prophase-I of meiosis is one of the longest and the most complex stage of meiosis. And the prophase-I is classified into 5 sub-stages. One is leptote. Here, you know, in entire prophase you will see condensation of the chromosome takes place. That is, the chromatin threads condense to form chromosome. The chromosomes appear beaded due to the presence of chromomeres. And in leptote, it has been observed that the chromosome forms a bouquet-like structure, spreading at one end and cluster at the other end. Zygote or synaptote. Pairing of homologous chromosomes starts, which is called as synapsis. Pair of homologous chromosomes are called bivalents. You know, each chromosome is univalent and homologous chromosomes are bivalent. There develops a structure between homologous chromosomes which is termed as synaptonymal complex. And here I may tell you one thing, that the pairing of the homologous chromosome is a highly precise process. It starts from one end and proceeds toward the other in a zipper-like fashion. Synaptonymal complex is composed of 3 thick lines of DNA and proteins. According to Moses, 1956, synaptonymal complex helps in the pairing and chiasmata. Chiasmata formation is the result of crossover. Here you can see the pairing of the chromosome. It can start from one end or it can start from the centromere. Pachytene. For our convenience, we will divide the pachytene into early and late. In early pachytene, you know, condensation will continue till dikinesis. Longitudinal splitting of the chromosome takes place. So, each chromosome becomes bivalent and homologous chromosome becomes quadrivalent. Now, in late pachytene, non-sister chromatids of bivalent develop recombination nodules and exchange their parts. That is, the crossing over takes place. And one thing I may tell you here, that the exchange of genetic material between two non-sister chromatids of the homologous chromosome never exceeds beyond 50%. Now you can see the exchange is within the limit of 50%. Theories of crossing over. Many theories have been given, like copy choice theory by Belling, breakage first theory by Muller, strain theory by Darlington, and breakage and reunion theory by Stern and Huta. And this is one of the most acceptable theory to explain the mechanism of crossing over. Breakage and reunion theory. And here you can see 2 enzymes are involved. You will see one is which breaks the non-sister chromatids, and then the ligase which helps in uniting these segments. Endonuclease enzyme first break the non-sister chromatids at the place of recombination nodule. Non-sister chromatids reunite by exchanging their parts with the help of enzyme ligase. As the result of crossing over, you will see a structure which is called chiasmata are visible. You know, the number of chiasma formation depends upon the length of the chromosome. It may be one, or if the chromosome is long, you may see it may be more than one. Next is the diploid. Condensation continues. Homologous chromosomes start repulsing each other, so chiasmata becomes visible only in diploid, and due to repulsion, chiasmata starts opening or sliding in a zip-like manner, and that process is called terminalization. And terminalization starts at diploid and will be over by the time it reaches the next substage, that is the dikinesis. Here you can see the diploid, and you can observe various shapes, loop, X-shaped, etc. Dikinesis, terminalization is complete. Homologous chromosomes remain attached to each other only at ends. Centrioles move to opposite pole, nuclear membrane disappear, and nucleolus disorganize and forms a matrix around the homologous chromosome. And in addition to that, as you know, that dikinesis is considered to be the best stage for counting the chromosome number in meiosis, because by the time the cell reaches the dikinesis, maximum condensation of the chromatin material has taken place. You can see the various loop-like or X-shaped structures are formed. Metaphase-I. The homologous chromosomes come to lie at the equatorial plane in such a way that the homologous chromosomes are at equidistance to their respective poles. Spindle fibers originate, and they are of two types. One is half-length fibers, that is from pole to centromere, and another is full-length fibers from pole to pole. You can see here Anaphase-I. The movement of homologous chromosome to their respective poles is the beginning of Anaphase-I. Chromosomal fibers contract and interzonal fibers expand. Anaphase is characterized by segregation or disjunction of homologous chromosome. You can see that the homologous chromosome starts moving towards the two poles. And again, the same push and pull mechanism is involved here also, as in mitosis. Telophase-I. Just like the telophase of mitosis, and here you will see that the nuclear membrane reappears and the nucleolus reorganizes, and you can see the two daughter nuclei, and by the time the telophase-I has reached, the chromosome number has reduced to n from 2n. Cytokinesis-I. In animal, constriction and furrow formation, as you have seen in mitosis; in plant, by cell plate formation. In most of the plant, cytokinesis does not occur after meiosis-I. It occurs only once, that is after meiosis-II. Group of 4 cells produce simultaneously, and it is called as tetrad. Tetrad of pollen grain, maybe of megaspore, and gap between meiosis-I and meiosis-II is called as interkinesis. Now here you can see the two daughter nuclei, means meiosis-I is complete and a constriction is arising. Of course, it is a case of animal cell. Preparation of meiosis-II occurred during interkinesis. It cannot be termed as interphase, as replication of DNA does not occur in interkinesis. Meiosis-II. It's a homeotypic division or equational division or ordinary mitotic division. It does not lead to any change in chromosome number. In meiosis-II, mother cell is haploid, daughter cells are also haploid. Meiosis-II is an ordinary mitotic division. In meiosis, division of the nucleus takes place twice, but the division of the chromosome occurs only once, and that is during the meiosis-II. You can see meiosis-I resulting in two daughter cells, and then the meiosis-II results in the formation of 4 daughter cells. Condensation of chromosome does not occur in amitosis. It is a process of division without recognizable chromosome. Amitosis does not involve the formation of spindle fibers. Here in amitosis, the division of the nucleus is direct, that is without sequential changes, that is the division into prophase, metaphase, anaphase, and telophase stage. In amitosis, division of cytoplasm and nucleus occurs simultaneously and by the method of constriction. This is, you can say, the mitotic division. 2 diploid cells are formed. Amitosis is the cell division of prokaryotes. But in certain eukaryotes like budding yeast particularly, where you will find that amitosis is occurring, and this is a budding yeast.